Howdy, my name is Angelica Ramirez, and today I'll be presenting on our bacterial staining lab report. In this experiment, the different types of staining methods were explored in order to distinguish between the microorganisms provided. The simple staining method will show the shape and arrangement of the bacteria. The gram staining method helps in differentiating between a gram-negative bacteria and a gram-positive bacteria. The acid-fast staining identifies acid-fast bacteria. Endospore staining detects endospores in the bacteria, while capsule staining is used to visualize bacterial capsules. For our simple staining, we were using a compound microscope, SRS blood agar plate, B-Series blood agar plate, S. typhimurium blood agar plate, our unknown cultures 103 and 203, a wax pencil, inoculation loop, deionized water, glass slides, methylene blue stain, and an immersion oil. First, heat-fixed slides of SRS, B-Series, S. typhimurium, and unknown cultures 103 and 203 were prepared. And to do this, small bacterial samples from solid agar media were collected and combined with a small drop of water that was placed on a clean slide beforehand. These samples were allowed to air dry before heat fixing over a flame. In order to heat fix it, the slides were passed briefly over a flame 3 times, and once the slides were heat fixed, they were then placed on the staining tray and flooded with methylene blue stain for 1 minute. The slides were then rinsed with deionized water and observed under the microscope. For our gram staining method, we used a compound microscope, S. typhimurium, SRS, B-Series, mixed culture, our unknowns 103 and 203, a wax pencil, inoculation loop, deionized water, glass slides, crystal violet, grams iodine mordant, safranin, immersion oil, and our decolorizer. The slides for the bacteria and solid agar media were prepared as before. For those grown in broth media, an inoculating loop was used to transfer 2 to 3 drops of the broth into a clean glass slide. The prepared slides were then allowed to dry before heat fixing. Once all the slides were prepared and heat fixed, they were placed on the staining tray and flooded with crystal violet stain for 1 minute. The slides were then rinsed with water for 5 seconds before being flooded with grams iodine mordant. The stain was left to stand for 2 minutes before rinsing the slides again with deionized water for 5 seconds. Acetone was then used to decolorize the slides before immediately flushing with water for 5 seconds. Safranin stain was then used as a counter stain and allowed to stand for 1 minute before rinsing the slides with water and blotting them dry with bubbleless paper. The slides were then examined under the microscope. For our acid-fast staining, we used a compound microscope, a prepared slide of Mycobacterium, wax pencil, deionized water, carbofusion stain, immersion oil, methylene blue, and acid alcohol decolorizer, and bubbleless paper. The pre-prepared slides of an acid-fast positive and acid-fast negative bacteria were used for the staining technique. The slide was first flooded with carbofusion stain and allowed to stand for 2 minutes. The slide was rinsed with water prior to rinsing it with acid alcohol decolorizer for approximately 3 seconds. Deionized water was used to rinse any residual decolorizer before flooding the slide with methylene blue stain for 30 seconds. The slide was then rinsed with deionized water 1 last time before being blot dry with bubbleless paper. The samples were then viewed under a microscope. Our capsule staining, we used a compound microscope, Kinemoniae, a wax pencil, deionized water, crystalline violet, inoculation loop, 2 glass slides, acid alcohol decolorizer, and bubbleless paper. To prepare a slide for the capsule staining, a small drop of crystal violet stain was placed on a clean glass slide, and then a loopful of K. pneumoniae was then mixed into the dye, being careful not to spread the stain. A second slide was then used to drag the stained cell mixture into an even thin film along the first slide. This was allowed to air dry before using the microscope to observe the sample. For our endospore staining, we used a compound microscope, B subtilis, a wax pencil, inoculation loop, deionized water, a glass slide, malachite green staining, safranin, immersion oil, and a Bobilis paper. First, a heat-fixed smear of B subtilis is prepared on a clean glass slide. The slide was then placed on a staining rack and covered with a filter paper. Malachite green staining was then used to saturate the paper. And then the slide was heated over a flame for approximately 5 minutes. If steam began to appear, it was set aside and allowed to cool before returning to the heat, while ensuring the filter paper didn't dry out. After 5 minutes, the filter paper was removed and the slide was rinsed with deionized water for 30 seconds. Safranin was then used as a counterstain and allowed to sit on the slide for 1 minute. After 1 minute passed, the stain was rinsed off and the sample was observed under the microscope. These were the results for a simple staining. We can see SRS, Bceres, unknown 103, 203, and S. stifamerium. The simple staining technique let us visualize the shape of present bacteria, but it didn't give us any other information. It is easy to identify the shape of Bceres, S. stifamerium, SRS, and unknown 3. However, a high presence of bacteria in the unknown 203 sample obscures any clear classification. Bceres is bacilli-shaped, unknown 103 and S. stifamerium were also bacilli-shaped, while unknown 203 and SRs were both cocci-shaped. These were the results for a gram staining. The mixed culture had a mix of gram-negative and gram-positive. Our unknown 103 was gram-positive bacilli, a Bceres was a gram-positive, SRS was gram-positive, S. stifamerium and unknown 203 were both gram-negative bacilli. In addition to the identification of the cell sample, or the cell shape, gram staining gives us a specific information about the makeup of the cell wall. Cells with a thin cell wall and outer membrane will stain pink, while gram-positive bacteria with a thick cell wall will stain purple. Our unknown 103 Bceres and SR seem to be gram-positive, while S. stifamerium and unknown 203 were negative. Both gram-negative and gram-positive bacteria were visible in the mixed culture sample. These were our results for our acid-fast staining. We could see a positive acid-fast and the negative acid-fast. Acid-fast staining is used to identify gram-positive bacteria with mycolic acid present on the thick cell wall. Bacteria that are acid-fast positive will appear pink, while acid-fast negative bacteria will appear blue. These were our results for capsule staining on K. pneumoniae. Because some bacteria are surrounded by capsules made of polysaccharides, it can be difficult to stain these particular bacteria with acidic stains. In order to visualize K. pneumoniae, an acidic stain was used to stain the background and a basic stain was used to stain the cell itself. It was quite difficult to see the clear halo that is normally visible during this type of staining in our sample, though. This was our endospore staining on B. subtilis. Endospores are bacterial coverings that are highly resistant to damage from extreme environmental conditions or chemicals. This makes it difficult to stain endospores by normal staining techniques. In order to encourage absorption of the stain, the B. subtilis sample was held over a flame. The endospores in the sample appear green under the microscope when stained successfully, which we did. All staining methods used in this experiment were very useful and helped identify the bacterial morphology, shape, and bacterial characteristics each of the different bacteria had. The simple staining method helped us know that B. cereus, unknown 103, and S. diffamerium are all bacilli-shaped bacteria, while unknown 203 and S. aureus are both cocci-shaped. Gram staining helped us see that unknown 103, B. cereus, and S. aureus are all gram-positive, while unknown 203 and S. diffamerium are gram-negative. In the mixed culture, there was a visible gram-positive and gram-negative bacteria. Acid-fast staining helped us verify whether the bacteria tested was acid-positive with a pink color, while if it was acid-fast negative, it was a blue color. In the capsule staining, K. pneumoniae was a bit difficult to see the capsule surrounding the bacteria, but in the endospore staining, we were able to see present endospore stained color green amidst the B. subtilis. Understanding the importance of the staining methods in the microbiology is very important, as it helps us understand and explore the microbes in a very thorough way that is very important in the medical field nowadays. And this concludes our lab report for this week. Thank you.